All right, welcome everybody. This is the Virtual Science Lab, defining cell types by their electrophysiology. I'm gonna go ahead and share my screen. All right, so our agenda today, it's now 10:30. My name is Caitlin, and I'm going to be doing a brief introduction for you to the Allen Institute and where to find our resources for educators. Then I'm gonna turn it over to Tom, who is gonna do an overview of some selected resources available at brainmap.org that are used in our featured lesson today, defining cell types by their electrophysiology. And then Ashley will demonstrate how to use that lesson in virtual experiment in your class. We will have plenty of time for questions. So please go ahead and put those questions into the Q&A button on Zoom or into the chat if you are joining us on YouTube. If you're watching the recording of this after the fact, there'll be a link in the YouTube description where you can go ahead and follow that, see the questions that were asked live, and ask your own questions as well. So participants, please make sure that you are sending chat messages to all panelists and attendees so others can see your message. If your Zoom name isn't your actual name, we'd love to know who you are. So please send a private message to moderator Christina to have yourself renamed. And viewing closed captions is actually by clicking the link that I just sent in these Zoom and YouTube chats, not the closed caption button. And that will go for the recording as well. Our presenters today are Tom and Ashley. Tom is a scientist in the Allen Institute for Brain Science who works on the data that is used in this open lesson plan. And Ashley is an assistant teaching professor at the University of California, San Diego. They'll both introduce themselves a little more when they speak. The Allen Institute is a biological sciences nonprofit located in Seattle, Washington, with focused research areas in neuroscience, cell biology, and immunology. We also support cutting-edge research across the world with the Paul G. Allen Frontiers Group. Across our institutes, our scientific process is focused on tackling complex, broad, and hard problems in the fundamental biological sciences. And this approach is guided by three principles, which I'm going to return to in a moment: big team and open science. And our work generates data, knowledge, and analysis tools, all of which we share publicly. To date, we've collected about 8 petabytes of data. That's 1.7 million DVDs worth. So these three core principles are team, big, and open science. Team science means that we work in large interdisciplinary teams that bring many different angles of expertise to a common complex problem. Big science is that we generate these large data sets that have robust, comprehensive data. And the most relevant today is open science, which we share all of our data for you to use in your research or with your students. So you can find our neuroscience data at brain-map.org and our cell biology data at allencell.org. So most of our resources were developed with research applications in mind, but they can be used in classrooms to provide students with genuine research experiences without the need for access to lab facilities that might not be available in all schools or while distance learning. We recognize that high-speed internet is also far from universal, but for many students who are safe at home, virtual experiments can provide a bridge to continuing their science education when you can't get into a lab in person. And even when you're back in your classrooms, we hope that the open data and the coding resources can be a resource for studying human cells and the brain by providing access to types of data that typical high school and college teaching labs just don't have the facilities or time to collect, or to expand your data sets so you can analyze more data and get more robust results. So to get started for educators, there's 2 places to head to find resources for educators. alleninstitute.org/learn will take you to lesson plans and posters for when you're back in your classroom. And this includes resources for teaching both neuroscience and cell biology. We have more resources coming soon as well. At allencell.org, you'll find links to cell biology specific data resources if you also teach general biology or want to share it with your colleagues who do. And you can view recordings of past webinars for educators at alleninstitute.org/thevirtualsciencelab. You can find the featured lesson for this webinar at alleninstitute.org/learn, and then scroll down to find the lesson plans section. So check out the other lesson plans that are available there as well. So I am now going to hand it over to Tom. As a reminder, please use that Q&A button down in the footer of Zoom to ask questions of our panelists. I'll share any immediate requests for clarification with them and then hold most of the questions until the end. And then you can use the chat to chat with each other, talk about what you're doing in your classes, and share any other comments that you have. And please remember to send those chat messages to all panelists and attendees. All right, Tom?
Thanks, Caitlin. How's that look? Slides good?
Looks great.
Great. So hi everyone, good to be here. Thanks for joining us. My name is Tom. I'm a scientist in the Human Cell Types program at the Allen Institute, where I've been for about 2 years now. My particular focus is on data analysis and modeling, but I work closely with the experimental teams generating this data as well, and I'm really excited to share it with you. So I'm going to first introduce the resources available in the web resource of the Allen Cell Types database, and the background there. It'll really be closely tied to Ashley's lesson. And then I'll also explain some of the more cutting edge work going on in the cell types world, where we're trying to infer cross-modality cell types in mouse and human. Some stuff I'm actively working on, and which will also represent future public resources. So I won't be saying much about how to access those yet, since they're not quite there, but feel free to ask me in the Q&A if those pique your interest. So what is the cell types database? It's a research program, one of the more recent ones here at the Allen Institute, started in 2015, and shifting our focus from more previously looking at large-scale anatomical atlases across the brain of mouse, to focusing down at the properties of single cells in mouse and human cortex, using those single cell properties to infer cell types. All of the data from this effort is released online with user-friendly access tools, so you can browse it and visualize the data in your web browser. But open science also means we share more than just the data. So scientific publications, of course, we try to make open access if possible. And we share the research tools used to generate this data, especially the software used to run these analyses. And so actually we'll be working with one instance of that, but there's many other packages we've released on GitHub that are available for use educationally or in research, of course. And we also release biomaterials. So this is important for researchers. We value sharing all the tools we use, including transgenic mice and viral tools, et cetera, all available. What is a cell type? This is an idea of classifying cells into groups that's been around since the early days of neuroscience, of course. Fundamentally, we just want to, it's useful to describe a cell type when we have a distinct population. So this could be a group of cells that is discontinuous with other groups in the sense of having properties that are seen in one and not seen at all in the other group. It could also be that another perspective on this is that cells are more like others in the group than cells outside the group. This reflects that we hope that the categorization, so saying a cell belongs to type A, helps us infer other properties. So inferring that other properties that we haven't yet measured would be similar. For instance, this also allows us to draw insight from other work that with the same cell type, and hope that those properties remain similar across research, across labs. So to be a little bit more concrete, we want a group of cells to share features obtained by one or more measurable criteria. This could be cells that have similar morphology, cells that carry out a similar task, and ideally we'd like these assessments of similar morphology or similar tasks to be quantitative and linked to function both. This is a pretty high bar, but we think both these aspects are very important. Quantitative aspect is important because it permits reproducibility, of course. So we'd like these definitions to be not just quantitative, but ideally, encoded in publicly released code that other researchers could rerun themselves and obtain exactly the same results and use these same type definitions. We wanted these definitions to be linked to function because this isn't just an empty exercise in labeling types. We want it to be useful for understanding the brain. And so if it's linked to function, we think it will be, just as if we're understanding a circuit, we build a parts list and then a wiring diagram and build towards that functional understanding. Or if we're understanding an ecosystem, we first distinguish species and then build up a food web to understand how those pieces interact. Similarly, we hope we can build on these definitions of cell types to reach a functional understanding of brain systems. Another fundamental principle in our understanding of cell types is that classification into a type should be flexible. Different definitions may be appropriate for different purposes. One aspect of that is to have hierarchical types. So for some purposes, distinguishing at a very coarse level of cell class, that's the term we use for just saying whether a cell is excitatory or inhibitory, might be appropriate. For other applications, we might go to the subclass level, which we often use to refer to the classical distinctions of, say, parvalbumin versus somatostatin interneuron types. But the level we'll focus most on, and which we use the label cell type for, is a fairly fine level of distinction that we usually inform by transcriptomics or by tree driver line. I'll say more about that as we go on. Now, in addition to having multiple levels possible in a hierarchy, we also think that regardless of the level, we should present this as a testable hypothesis. It's not known a priori that all variability in the brain should be well organized into types. It could be random variability. And so we want to present the evidence to users like you and let you make your own calls of whether you think this is a meaningful type distinction. And again, this is partly facilitated by having these open tools release available so you can see the evidence yourself. So as I said, cell types are a very old concept, but there are some new aspects that make this an ideal time for us to start the Allen Cell Types Database enterprise. So historically, definitions of cell type were often fairly coarse or qualitative, not as reproducible, because they relied on aspects like morphology, so recognizing the distinctive morphology of a particular type in stained tissue. Sometimes this works great for highly distinctive cell types like G cells or chandelier cells. It's very hard to miss them. But in other cases, it relies really tightly on expert knowledge and is harder to reproduce for that reason. There's also immunohistochemistry-based identification of cell types like parvalbumin or somatostatin interneuron, for instance, which is great. Again, that's highly reproducible, but it's fairly limited. It relies on a small set of marker genes rather than the full array of potential gene expression distinctions that we could use potentially to identify cell type. What do you have available to us now? Well, on the transcriptomic side, we have next generation sequencing tools and sequencing at the single cell and single nucleus levels from bulk tissue to allow us to get gene transcript data for extremely large numbers of cells, so thousands or tens of thousands of cells very easily. We also have transgenic technologies and tools that allow us to target specific cell types that might be so rare they would essentially never be found in random sampling of tissue. We have high resolution imaging and image processing that lets us get really detailed morphology like these shown here. Not just get images of them, but also process those into data which we can extract low dimensional features from. I'll say more about later on. And we have the computational power to run these analyses, especially important on genomic data, to run high dimensional analysis and visualization, those to better understand our data. Coming with all these new opportunities, we also have renewed appreciation and funding for this descriptive endeavor. So a lot of the work I'll be presenting here is made possible not only through Allen Institute funding, but through grant funding as well, and that's important. What do we bring to the table in this modern look on cell types? Well, our goal is to use our open science and big science approach to scale up, refine, and standardize existing techniques into a pipeline. Pipeline just reflecting that high level of reproducibility and rigor in our work. And again, is made possible by looking broadly for available tools and then refining them, using our in-house expertise. Our goal is to go deep, so aim for a CAP data set, CAP standing for complete, accurate, and permanent. We want this to be a tool that's useful long into the future, and so we want to be very careful up front to extract everything possible, make it as useful as possible for scientists in the future. Okay, so with that that background in mind, I'm going to first start by explaining what's currently available at celltypes.brainmap.org, our web resource. So this subset of the cell types program we call the in vitro single cell characterization pipeline. So studying the properties of single cells in vitro and focusing on morphological and electrophysiological properties, or morphoelectric. Ashley will be looking at in her lesson as well. So this comes from data from two model organisms. So we have a large mouse colony in house that we use to generate tissue that's spliced and then recorded, and we're able to record from single cells. We also have unique collaborations with Seattle area hospitals, where neurosurgeons who are carrying out brain surgery for epilepsy or tumors with consent of the patients in some cases are able to get very small chunks of tissue that need to be pulled out during that surgery anyway, and send them over to the institute fast enough that we're able to keep them alive and record from them the same way as we would with mouse tissue. So in either case, once we have that tissue in the lab, the first step is to target specific cells for whole cell recording. So that's a whole cell patch clamp recording, and in which we inject different stimulus protocols, which I'll talk about in a moment, to test the cell's input-output properties. Following that recording, we inject the cell with dye, and then once all the recordings from that slice are done, we image it and process that image to create a 3D reconstruction of the cell, meaning that we actually represent the cell as a set of points and lines that can be analyzed using software to extract properties like the length or width of the cell or branching properties, etc. And also to generate models. On the EFIS side, we take these raw recordings, these traces of injected stimulus and response of the cell. And again, we process those traces, which I'll talk a bit more about, to get a large set of features that capture both the single spike properties as shown in this illustration and the other types of response. And these are two categories of resource that are available at celltypes.brainmap.org. The third is models, computational models that we design to reproduce either just EFIS only in the case of the simpler models, or in the case of more complex models, they include both the morphology and the EFIS. So I'll say a bit more about these categories. I'm not going to talk about them in depth, but these are also available to visualize on the website. You can see whether or not these models are able to reproduce the input-output properties of cells. In the cases where they're good, where they are accurate reproductions, we think these are a very useful way of understanding the variability and what the underlying factors behind variability might be. Okay, so the first thing you see when you get to this part of the website is, a very detailed selection of sample metadata that you can use to target cells of interest, for browsing. If you have a particular type or brain region or species you're interested in, that's all available. So again, brain areas, layers, hemispheres, all this metadata is annotated and able to use to select subsets. So a few to highlight here from the cell types perspective, the important aspects that we might use to distinguish cell types are on the mouse side, we use transgenic Cree lines to target putative cell types. So some of these might be very broad, like all inhibitory cells. Some of them might be more specific, like a parvalbumin specific in cancer. And this can be selected for browsing by just using this transgenic targeting selector and making sure you also select report positive. A broader level of cell type, or cell class rather, would be for cells that have morphology. You can select spiny or aspiny dendrites, reflecting that cells with spines in the dendrites are typically excitatory and aspiny typically inhibitory. So more detailed level of cell typing driven by the data I'll come back to later on, but there are levels we can address just by the metadata and selecting a subset of that data available. Okay, so what do we record from these cells? So we have a standardized set of stimuli that are input to every single cell in the pipeline. in the form of a consecutive series of sweeps. A sweep being some cases very short, like a single pulse that triggers a single spike in the cell, some cases much longer, like these ramps that are over several seconds, or a single pulses over a second. These are designed to interrogate the input-output function of the cell, and also to be suited for calculating features, so a lower dimensional representation of that response. We also have some more complex stimuli that are designed to more closely mimic the complex activity that the cells are responding to in vivo. These include a short, precisely timed series of spikes that we can see how the cell responds to, and noise stimuli overlaid on either a ramp or long squares. And since these are more closely mimic in vivo activity, we think these are better for constructing and validating the models we're generating. They're primarily used for that rather than generating features of cell typing. So for the first set of stimuli, the feature generation ones, the step following recording is to take those recordings, the data, and run some analysis packages to extract features that summarize the distinctive characteristics of each cell's response. So there's several categories of features we use. There's superthreshold features, so reflecting the spiking properties of the cell when the stimuli is strong enough to make that cell fire. And these could be features of a single action of potential, like the voltage threshold it starts at, upstroke downstroke rates, positions of the peak and the trough following the spike, etc. There's also features at the level of the train of action potentials that rely mostly on the timing of those spikes. So how the interspike interval separating the spikes, or the ISI, how variable those ISIs are, how long it takes for the first spike to happen, or the latency, etc. There's a number of properties that rely on the spike times and are calculated for every sweep in the data set. Then it's also important to assess the properties when the cell is not spiking through subthreshold current input, either hyperpolarizing input or depolarizing input that's below the threshold for the cell to spike. So we use those to test resting membrane potential, input resistance, time constants of the membrane, the sag in response to hyperpolarization, etc. I should say we're always working on adding more features that better inform the analysis of cell types. A common set is, and a more basic set is available in what actually we'll be showing, but we also released the more cutting edge tools in a newer analysis package called IPFX, which is also publicly released. So back to the web resource, again, that final component that we have on there are EFIS models, or models for both EFIS and morphology in some cases. So this Venn diagram shows that this is only really available for a subset of the data. So these more stereotyped abstracted models, known as the generalized leakly integrated fire model, rely only on EFIS, and so these are available for quite a bit of the data. Then we have more detailed levels of biophysically detailed modeling, which rely on actually simulating the structure of the cell and what the voltage is at different compartments of the cell, as well as how the specific ion channels involved to determine those voltage dynamics. The most recent release there, so just a paper available on bioRxiv, are what we call all active models, which have active ion channels in the dendrites as well. So those are the smallest set available, but the newest. And if you have interest in those models, feel free to ask questions later on. Okay, so I'm going to shift gears a little bit here now and move into some results that are recently published papers from the cell types program, but not yet really available in the online resource, in which we use these cell features to determine more data-driven cell type distinction. So there's a variety of ways we can do this, since we have several different modalities of data. Of course, so I've already talked about the morphology data from those 3D reconstructions of biocytin-filled cells, and the electrophysiology data, which Ashley will show more about from whole cell patch clamp. In both cases, I'm going to be assuming we've reduced those complex data, as shown here, down to a small set of features, so lower dimensional representation. representation. For most of the cells, the newer iteration of the pipeline, we're also running a patch-seq protocol, meaning that following the patch clamp recording of the cell, we extract the nucleus and sequence the RNA in that nucleus. That allows us to determine what genes are currently being expressed in the cell, detailed picture of cell type from that perspective. We also carry out single cell sequencing of bulk tissue that we don't have the other modalities available for, which lets us, for transcriptomics alone, get really large scale data sets in the tens or hundreds of thousands of cells. So with each of these modalities alone, we can run different varieties of clustering procedures to create property-specific taxonomies in each modality. And so if we run a clustering on morphology data and determine several groups of interneurons as shown in this illustration, we call those M types, so morphology-driven types. We can do the same for E types. And again, this clustering shows that we can distinguish several islands, as we might call them, in this low dimensional representation that are extremely distinct and from the others, just determined by those EVIS properties. We do the same thing with transcriptomics, and again, because this is where we have the most data, this is where we can also get the finest resolution of typing in a rigorous way. Because in this case, whereas the M and E types are more 10 or 20 inhibitory types, as shown here, more like in the hundreds for the T types because of that data availability. However, it's not really satisfying to have three different ways of assigning cell types. So the biggest challenge here is how to integrate across these different modalities and decide what a principled way of creating multidimensional cell types is. So our first step at this was to assign ME types simply by aggregating together, creating a combined space of morphology and electrophysiology, and using that to run clustering. And since it's more data than morphology or EPIS alone, it allows a somewhat finer level of distinction shown here than the previous page. But integrating that with transcriptomics is challenging. This is really the cutting edge here now that's only available, that's currently a preprint on bioRxiv. Our current approach to this is to sort of create a consensus, take the existing ME types and align them to the T types at single cell level, and then look at where they are compatible and where they are not, and focus on types where all three modalities are compatible, to use that for our best call on cell type. This brings us back to a slightly smaller level because a lot of types get merged in that process across the modality. The result for SST cells only is shown here. Now, those results I showed previously were all from mouse data, where we have thousands of patch-seq cells, patch-seq interneurons. On the human side, which is my focus, we have much less data availability. So we are faced with unique challenges there, where we can't quite, at least yet, we can't run that large-scale MET clustering approach. So here we rely even more heavily on the transcriptomics. So we are able to get about, order of 15,000 cells sequenced from human tissue, and that's enough to create a very detailed cell type taxonomy. So again, you see here in this low dimensional representation of transcriptomic space that there are many type distinctions that are highly discontinuous and highly rigorous in that sense. But from the patch-seq data, the patch-seq data is a much smaller data set. So that's more on the order of several hundred, 400 cells that so far we've looked at in a layer 2-3 data set. So that represents sort of a subset of this cross-layer, cross-type view over here, zoomed in. And the way we deal with that smaller data set, it's not enough to cluster on its own, but we can take advantage of the larger sampling from transcriptomics only and align the smaller data set to the larger. So you see how they're overlaid in the same space here, a subset of this large space. And in that, once they're aligned, we can use the larger the taxonomy from the transcriptomics to assign types to the patch-seq cell. Those are shown here. These are shorthand cell type labels based on marker genes for each type. These are 5 types that are found within excitatory cells in layer 2-3 of human tissue. Once we have those T-type labels, our approach is to use those to help understand the diversity in the morphology and the electrophysiology data. The T-type labels help permit cross-species comparison, since we have alignment between mouse and human T-types, can help us understand how types vary over evolution. We can also look at whether that diversity is correlated across modalities, whether types that are distinct transcriptomically are also distinct morphoelectrically. The results from this are pretty interesting. So this is data just released as a preprint also. We find that compared to mouse, the excitatory cells in layers 2 and 3 show much greater diversity in a few senses in human mouse, which is it's expected since there's expansion of these layers in particular through evolution. Primates and humans in particular have much larger depth of layers 2 and 3. And we see both distinct types. So there are these lower types, T-types in this picture are ones that are more similar to T-types found in layers 5 and 6 of mouse cortex, but they're part of the diversity in layers 2 and 3 in human, which we think is interesting. We also find that some of these types, so from this from3 labeled type in particular, are found throughout the depth of layers 2 and 3 in human, and very strongly in their properties throughout that depth. So they have that very within type variability that's not really found in mouse. So in these two aspects, and also, more in depth, like in the paper, of course, we think that the cell typing, so assigning those T-types, facilitates comparison both between types and across species, and by doing so moves us closer to understanding the circuits of layer 2-3, which of course is our ultimate goal. That's the end of my part here, just a reminder that all these results are possible thanks to the generosity of our institute founder, Paul G. Allen. And all the results I'm showing are, result of team science. It involves so many people across the institute, across different teams that work together closely to make this happen. I can't list them all here, but they're all critical to this. And of course, this is also grant-supported work in much of what I'm showing here, supported by different grants here. So let's move to Ashley now. Thank you.
All right, thanks, Tom, for that great introduction. So hello, I am Ashley, and I'm going to be telling you a bit about a lesson plan that you can use in your class to actually teach some of these materials and resources that Tom just introduced us to. So hopefully you can see my screen okay. So I'm an assistant teaching professor at UC San Diego, and I wanted to give you a little bit of background about where I'm coming from, so that hopefully this can give you some confidence in actually using some of these materials. So I came from a small liberal arts school on the East Coast at Lafayette College, where the coding class was optional and I didn't take it. In my PhD, I quote unquote learned how to code, which for many grad students means I was given code that other people had written in MATLAB, and I dabbled a little bit in Python. As a postdoc, I did more MATLAB. And then it wasn't until I joined the faculty at UC San Diego just over 2 years ago now that I actually learned Python. So I'm fairly new to Python, and hopefully that means that you too can actually teach Python in your class, even if you've never taken a formal coding class. So first bit of inspiration for you this morning. So the first thing I want to do in this webinar is to provide a general framework for how you can actually teach coding in your class, which sounds like for many of you is a neuroscience class or a biology class. And then what I want to do is actually demonstrate the use of Jupyter notebooks to analyze some of the data that Tom just told us about. And so we're going to do an interactive demonstration. So hopefully you came ready to code this morning. So first things first, I have a question for you all. We're going to launch a poll in just a second. So the question is, do you teach or demonstrate programming in your courses? And so the options you'll see will ask you if you teach or maybe just demonstrate in your courses, or maybe you do neither of those things. So take a minute if you're here and go ahead and respond to the poll that you should just see now. All right. And whenever we have a majority here voted, we can go ahead and release those results. Awesome. Okay, great. So this is really good to know. So it looks like most folks are not using coding at all in their classes, and we have a few people who are demonstrating coding and a few people who actually do teach coding in their classes. So that's great. So I'm really happy to have some folks here for whom this might be totally new information, and that's wonderful. So the first thing I want to do is actually motivate the need to teach neuroscience students how to code. And I'm not the first person to think of this. So there's this really nice review in 2016 by Bill Grisham and others about how neuroscience students need to learn how to work with large scale data as data gets bigger and bigger in neuroscience. There's another really nice article in 2018 that really makes an argument that students need these quantitative skills and familiarity with statistical applications and scientific programming. There's another paper down here by Akil et al. About training our students for the 21st century. And then I'll make a small plug too for the fact that I just published a book which also makes this argument. Neuroscience is changing and we need to invite our students, all students, into the pursuit to analyze some of this large scale data that we have. Yet few neuroscience programs around the country actually require coding and only some offer it as an elective. So the case I'd like to make for you this morning is that we can really easily integrate coding, even just exposure to coding in our neuroscience classes. And this is especially true in remote laboratory courses where we can very readily actually teach students how to code online. Of course, students are coming into our classrooms with preconceived notions about coding, especially biology students who may have already sort of made the decision that they're not a math person, they're a biology person. So it's really important to think about this before you introduce coding in your classroom. So students might think, my roommate is a CS major and those classes are really tough. They might also think, I'm not a math person, so I won't be able to code. A lot of people tend to conflate those two things. Or they might have preconceived notions about who codes. So they might say, coding is for rich white dudes. And so we have to acknowledge the fact that students have these preconceived notions before we can actually teach coding. So to think about this and to think about how to approach this with your students, the first thing is to say there's no programming gene, right? Someone wasn't born knowing how to code. Instead, coding is a skill. It's like learning a language. And related to that, it's really important to encourage growth mindsets around coding. And there's a lot of literature on why growth mindsets are important. So students with growth mindsets perform better over time, even in really tough classes like organic chemistry. And the belief about whether computational abilities are fixed or malleable impact a whole variety of things, including students' sense of belonging in a classroom, how they respond to difficulties, and ultimately their achievement. So this is incredibly important in our classrooms to frame this as something that students can learn. And here's a few additional things that you can do if you're using these lessons in your class. So you can live code in front of your students. I'm going to do that for you in a few minutes. It's a little bit terrifying, but it's okay to make mistakes. And in fact, that's really important for students to see. You can encourage pair programming, which is a technique that's used in a lot of companies where you have a student who is driving the code. So they're actually the ones doing the coding, whereas you have a navigator who is the one instructing what to do in the code. As I just mentioned, you can normalize errors in searching for answers. So if you're someone who has coded before, you're new to coding, you know that it is entirely normal to go to Google and say, how do I do X, Y, and Z? And that's just a total normal thing. It's also very normal to get errors and bugs in your code. So we have to normalize all these things for our students before we dive in. Okay, so with that, I want to actually demonstrate the use of Jupyter Notebooks to analyze the data that Tom talked about earlier. And if you're thinking about teaching coding in your classes, you might be thinking about, well, which programming language should I teach? And a lot of students will have this question as well, which programming language should I learn? Well, here's a few considerations to think about. First thing is the fiscal entry cost. So just how much does it cost? And tools like Python and R are totally open source and free. So that's a really big plus. And then there's the conceptual entry cost. So some languages might be more in English. So for English speaking students, this is a little bit easier in terms of the barrier to entry, whereas other languages like C++ are basically not readable in English. So it's really hard. So not surprisingly, the Allen Institute, given its attitude towards open science, has chosen to use tools that have low fiscal entry costs and have relatively low entry costs as well, conceptually. So there's a few different ways that you can actually use Python in your classroom. And we're going to use Python in the form of a Jupyter notebook. And so you can run these Jupyter notebooks a couple different ways. So today we're going to run it in a Google Colab notebook. Another sort of similar way to do this is with a tool called Binder. One thing you can do too, if you talk to the folks at your course or university, is you can actually set up what's called a Jupyter Hub. And so at UCSD, the way that it looks for students is they go to datahub.ucsd.edu, they log in, and then this gives us a whole environment where they can code, and that's already set up for them. So this is a really nice way to go if you and maybe other instructors are going to teach this way. And then finally, you can run these notebooks locally. So you can actually download a distribution of Python, such as the Anaconda distribution, so that students can just run this locally on their own computer instead of relying on an internet connection. So this lesson that I'm going to show you today is part of a set of lessons that can take maybe 2 or 3 days in your class, depending on how you want to space it out. So here's a possible workflow for these notebooks. So typically when I teach this, I give some sort of lecture and have a pre-reading on electrophysiology concepts and also introducing students to the Allen Institute, just like Tom just did. Then we'll do some sort of web-based introduction. And so there's resources for that on the website, and I'll point you to that in just a second. And then you might spend 1 or 2 sessions actually going through some of the coding notebooks after that. So to break this down a little bit further, in order for students to actually get something out of this lesson, it's important to cover some important background things. So this is actually really similar to what Tom covered earlier. So you might cover the idea that there are different cell types in the brain, and we can define them by their electrophysiology or their transcriptomics, et cetera. You should also probably talk about whole cell patch clamp recording, since the data that we're going to talk about today was collected using whole cell patch clamp. And then finally, you can talk to students about how you can target cells based on their genetic expression. So the use of Cree lines, as the Allen Institute uses. So that's the first step in these lessons. The second step is a web-based exploration of some of the data, similar to the interface that Tom just showed you. And if you're curious, you can find all of the resources to be able to implement that in your classroom here. And I think we'll put the link in the chat as well for you. So the web-based interface looks something like this. So cells can see some of these pre-computed features about the cell, such as the dendrite type. They can also see all of these recording sweeps. And then finally, they can look at the morphology of the cell to see what the cell actually looks like. So once students do that and they get some exposure to what this data actually looks like, it's a good chance to then dive in and look at this data programmatically using Python. So the first step in that is to learn a bit of the basics. So there's an introduction to Jupyter Notebook in the same folder with the notebooks we'll look at today. And so you're welcome to work through that, and that would be really great for your students to do as well. But then finally, we can actually dive into the data, dive into the software development kit using Jupyter in a similar way. And that's going to be our focus for today. So without further ado, let's go ahead and code. So we're going to put a link in the chat to this Colab notebook. So that's going to help us create a Python environment where we can actually analyze some of the data using Python code. So the link should be in the chat now. Awesome. So you can go ahead and click on that. And I'll give folks a minute to get situated here. So you can click on that link. I'll show you what it looks like here. And you'll get a page that looks something like this. So this shows you all of the notebooks that are in this folder. And as you might have noticed in the workflow earlier, there's 2 different notebooks here. So there's a coding light and a coding focused notebook. So the coding light notebook, as the name suggests, doesn't ask the students to do a ton of coding, whereas the coding focused notebook has just a few additional coding tasks to work through. And since we're all going to be expert coders this morning, we're going to do the coding focused notebook together. So you can go ahead and click on that when you're ready. So this is going to load up a notebook that is running online. It's running on Google's servers, and hopefully it'll get connected here. You'll see up in the upper right-hand corner, you'll get a green check mark, meaning you're all ready to go. And so this is a notebook that's going to help us define cell types by their electrophysiology and dive into some of the cell types data that's created by the Allen Institute. So you can see here, there's a few steps to the notebook. So what we're going to do today is we're going to set up our coding environment, and then we're actually going to skip to the end here to look at data from many cells. These steps 2 through 4 are a way of plotting the data that you can look at on the website, just so students can kind of make the connection that this is the same data. So we can import the same data from the website, we can plot the data, we can get it to look like the website. This gives students a chance to really see, okay, there's a connection here between what we did on the website and what we're doing in the notebook. But for today, we'll skip to step 5. The first thing is to actually get set up. And so since we're running this online in Google Colab doesn't immediately have the Allen SDK installed. So we have to install it. So this very first cell, so this is a code cell here. So it looks different. So the markdown text is just plain, whereas we have code cell with lots of different formatting for different keywords. You can run this cell with the upper left-hand corner. So there's an arrow pointing to the right. So go ahead and click that cell. This is going to throw you an error immediately because it's going to say this notebook was not authored by Google. It's from GitHub. Do you trust it? You can trust me. Go ahead and click run anyway so that we can actually run this cell. So when this goes for you, it's actually going to install the SDK. So since I have ran this earlier, it's already installed for me, but you're going to see a lot of messages pop up here with it actually going through the installation process. So I'll pause here to let that work for people, and we can see if there's any questions immediately that I can answer while people are getting set up here. And again, if you missed it, we're in the coding focused notebook that's on the link that was put in the chat. And we're just going to run this very first cell by clicking on the arrow to the that's pointing to the right up in the upper left-hand corner. And we're going to install the Allen SDK tools here, which just takes about a minute or something. So once this finishes, you're going to get a notice that says you need to restart the runtime. And so what it's going to give you, it might have scrolled down for you. So if you scroll back up to this same step, there'll be a button in this cell that will say restart runtime. And if you click that, it's not going to do anything, but it'll restart the runtime for you. It'll say something about removing the variables from the workspace. That's fine. You can click OK. And so once you've gone through that step, then you are ready to go. All right. So I'm going to hope that folks are on board here. So now we can move on to the next step, which is to get our environment set up with a few additional things. So whenever we're using Python, we usually have to import packages. Python is very much built on whatever packages you import. So what we're going to do here is just make sure that we have these packages. So you can just run this cell. It's going to do a check for you that your environment has these packages that we need. And once this is done running, it's going to give you a nice message here to tell you that these things are already installed, since Colab already comes with these packages installed. So now that we have our SDK and our packages installed, so that's all good, we need to actually import them into the environment. And so that's what this cell below is going to do. And in this cell, we also encounter our first real task. And so what this task says, and there's many of these throughout the notebook, this says, just like you did in the introductory notebook, import the NumPy module nicknamed as NP, and add a print message at the end that says packages imported so that you know the code ran. So you'll notice here, there's already a bit of code to import some of these other packages and configure our plots in high resolution. But what we need to do here is actually import NumPy, and we need this below. So something will fail below if we don't do this. So let's make sure that we import NumPy, and we can do import NumPy as NP. And we say as NP, that's basically nicknaming the package. So rather than referring it to this full name as NumPy every time, we'll just refer to it as NP. And then we're going to add a print statement below. So we're going to say print, and then we're going to say packages imported. And then we can run this cell. And by the way, you can run either by clicking this, or you can hit Shift Enter, which is a nice shorthand too. Okay, so similarly, we also have to import this cell types module from the Allen SDK. And we're also going to import some data too. So you can pull the data directly from the SDK. But since that's a little slow this morning, I'm actually going to have you clone the data from the same repository where this code lives. So the only thing you have to do here, as the task says, is to uncomment these two lines. So we're going to uncomment this first line that says git clone. And then we're also going to uncomment this line that says cd cell types lesson. So what this first line is going to do is it's going to clone the data that I've already saved for you, and then it's going to just move into that folder afterwards. So after you've uncommented those two lines, you can go ahead and run this cell. And you will not get this message, but it should be fine. All right. So now we can actually start working with the data. And as I said before, we're actually going to skip steps 2 through 4, since those are just going to work with the same data that we see on the website. So for the tutorial this morning, we're actually going to skip to step 5. So if you click on the left-hand side here, Colab usefully has this really nice table of contents menu. And so you can use that to actually go down and click on step 5, analyze pre-computed features. So we're going to hop down there. Okay, so step 5 here is going to actually help us get some of those pre-computed features that Tom was talking about earlier. So there's sub-threshold as well as super-threshold features. And so the first thing we're going to do is just get all those features for our cells. And you can just run this, and this is going to give us a data frame. So a data frame is a really useful way to work with data sets like this. It's created by the package pandas, abbreviated here as PD, as we saw above. And so this data frame looks something like an Excel spreadsheet, and you'll see there's different specimen IDs. We're just looking at the very top of the data frame here. And you can scroll over and you'll see there's all these different features for the cells. Features like the input resistance or the latency. And so all of these are different features that you could analyze as you're working with this data. There's a glossary linked here in case you're curious what any of these features actually mean. So you can click on that, just a Google Doc with some definitions there for you. And here's that nice figure that Tom showed earlier as well. So let's first look at 2 of these features. So we're going to look at the fast trough, so the voltage of the trough in response to a long square stimulus. And we're going to look at the ratio between the upstroke and downstroke of the action potential. And so what we did above is we created a pandas data frame. And so what we're going to need to do is actually take out columns of that data frame related to these 2 different features. And so the first one is this fast trough. And so fast underscore trough underscore, et cetera. So what we're going to do is we're going to refer to the data frame using this syntax. So we're going to say data frame, and that's exactly what we called it actually above. We can double check. So we called it data frame. That's just our variable name. And then we can go down and we're going to refer to the column of interest in single quotes. So this is our first task to really assign a variable here. So we're going to replace these ellipses and we're going to say data frame. And so since we want this fast trough to be our fast trough one, Google Colab is actually usefully going to allow us to autocomplete it. So we can just click the fast trough v long square to get that metric. And same thing for this one. So we're going to say data frame. And then similarly, we are going to get the upstroke downstroke ratio. And autocomplete here will be your friend. And we can click that. So now we're just going to assign 2 different variables, fast trough and upstroke downstroke, so that those will contain all of the data from these columns of the data frame. And you could run this cell. We didn't add a print statement, so you won't see anything. But when the cell's done running, you'll get a check mark here in the upper right-hand corner. Okay, so let's see actually if these 2 things that we pulled out are actually related in any way. And so this is our first plotting task right now. So we're going to actually use Python to plot a scatter plot where we're just relating these 2 different variables. So in order to do so, we can do plt.scatter. That's how we do a scatter plot in Python. And Colab's actually usefully telling us the syntax for this. So it tells us scatter x, y, and then there's a bunch of arguments we could give the scatter plot. But what we're going to put on the x-axis is actually that fast trough. Once you see something pop up and Colab is suggesting something to you, if you hit tab, it'll actually auto-complete it for you. And then we'll do upstroke, downstroke. And then if we run this, we'll get a scatter plot here. So we have our 2 variables here. So on the x-axis is our fast trough. So that's the voltage here. We could add a label. So fast trough. And y label for the other axis. And we'll just call that upstroke, downstroke ratio. Might be good to have some units. There we go. So clearly there's something going on here. These variables are related in some way. We've pulled out the depth of the trough as well as the upstroke downstroke ratio. And there seems to be some sort of positive correlation here. There also seems to be different clusters in the data. So it seems like there's all of these cells here that are clustered and all these cells on the bottom that are clustered. So what this next cell is going to do is actually going to color these dots, which are individual recordings or individual cells rather, by their spine type. And so what we're going to do here, all the code is already here for you. You can simply run it, is we're going to create the same plot, but now we're going to actually color it by type. So now we're coloring it by spiny and aspiny. And as Tom mentioned earlier, spiny cells tend to be excitatory, whereas aspiny cells tend to be inhibitory. So now you can see really clearly that spiny cells live up here. So spiny cells have a shallow after hyperpolarization, and they have a really high upstroke downstroke ratio. So you might wonder, as we're looking at these metrics, what do these mean in terms of the actual shape of the action potential? Like maybe it would be good to actually plot an action potential and look at the waveform of it to see what it looks like. So what we can do here is actually pull out some values here. So first we're going to pull out a cell that has a really high upstroke downstroke ratio to get a sense of what that means in terms of the shape of the action potential. So this is going to sort our data frames so that the highest ones are at the top. And then we're going to grab one of those high ones and plot the actual action potential to kind of zoom in on it. Okay, so this is telling us the specimen ID and the upstroke downstroke ratio. So this is really high, it's about 6. If you look at our plot, this is one of the highest ones up here. So it's probably one of these cells here. All right, so the next task is actually to first get the data for this particular sweep. So you can run this, and this should be fairly quick if the cloning above worked for you. If this takes a minute or two, that might happen, but it should be fairly quick. You might get a warning here. That's not a problem at all. Our sweep was still obtained. So basically, we just pulled out the raw data for this particular cell for a given sweep that I've hand selected for you. So this sweep 45. You could, again, go to the website and actually look at the same cell on the website. But we're going to look at it here programmatically. Okay, and now we can plot this. So what we want to do is we can use this plot function. And so you'll notice actually there's a lot of plotting tasks in this notebook. Not surprisingly, a lot of plotting is generating, or a lot of coding rather is generating plots. So first thing we're going to do is we're going to do plt.plot. So again, we're going to use our coding package that we imported above, our plotting package, and we're going to do x times y. And above, we actually created something called timestamps. So you can go up here. So we have our timestamps, and we have current and voltage. And so if we want to plot the actual voltage response of the cell, we can do voltage here on the y-axis. So we can do plot and then timestamps and voltage. So this gives us the whole sweep, right? So this is 8 seconds of data. It's kind of hard to see the actual shape of the action potential. So it might be useful to actually zoom in on this plot that we have here. So let's go ahead and add a line of code that says plt.xlim. So this is going to set the limit of the x-axis. And let's do it for, let's say, like 1.5 to 1.7. So we're going to really zoom in here. And so now we can pretty clearly see individual action potentials. And you'll notice the shape of them looks kind of different than a canonical action potential, actually. And so this is our shape of our action potential. So this shows you what it means for a cell to have a really high upstroke downstroke ratio. So you could do the same thing, and the task here below is to actually have students do the same thing, but for a low upstroke downstroke ratio. And so I won't do this with you now, but the basic idea that you can, the way you can do this is just change this ascending false to true. So that's, instead of grabbing a cell with a high upstroke downstroke ratio, it's going to grab a cell with a low upstroke downstroke ratio. And then you can run the same cells below and plot the sweep once again. All right, final task for today. And this is really kind of the bread of this, right? So we want to actually compare types in the brain. And usefully, as Tom pointed out earlier, a lot of different ways we can actually compare cell types. And so the way this is set up is so that you can choose your own comparison. So if you want to compare cells from different species, you can look at the column species. If you want to compare spiny versus aspiny cells, you can look at this column called dendrite type. And if you want to compare two transgenic lines, you can look at the column transgenic line. And so the first thing we can do is actually define the column name below. So we're going to once again assign a variable. We're going to remove the ellipses here, and we're going to assign it to one of these. So let's go ahead and do species. So let's say we want to actually compare mice and human data. So we can run this, and what this is going to print for us is the unique values in this column. And so this data set has Homo sapiens as well as mouse cells. So now what we can do is we can use these to actually subset our data frame based on these different cell types. And you could do the same thing for spiny and aspiny. So this will give you spiny and aspiny, also sparsely spiny. But basically, you're going to choose 2 of the values from here. So we'll do Homo sapiens and mouse mus musculus in order to actually create 2 different cell types here. So we will create cell type 1 is Homo sapiens and cell type 2 is mouse cells. So now what this cell is going to do is it's just going to go through the data frame and create 2 separate data frames for human cells and mouse cells. And it's also going to tell us how many cells are in these different types. So we have 413 human cells and almost 2,000 mouse cells. Now, finally, let's actually see if these cells are different for any of the pre-computed features that exist in this data set. So what we can do is we can actually change the feature here. So this just starts us off thinking about resting membrane potential. So let's go ahead and take out resting membrane potential for our 2 different cell types and compare them. And so the first thing it's going to do, and this is already set up for you, is plot a histogram. So we can go ahead and run this cell and it'll plot us a histogram. So reminder that cell type 1 was human, cell type 2 is mice. And perhaps not surprisingly, human and mice have pretty similar resting membrane potentials. So resting membrane potential is on the x-axis here, and this is the normalized number of cells. So if you wanted to, you could go back and actually change this for a different feature. And there's a list of features below that you might want to compare. So let's say we have some idea, maybe we think that human cells might be different from mice cells based on their time constant. So we could actually use this tau feature. So if we go above, we can instead change this feature to tau. We're going to keep it in single quotes because it needs to be a string, and run this again. And so now we can see, okay, this looks pretty similar as well. So maybe human and mice cells aren't that different. If you read the Allen Institute papers, though, there is at least one feature that's different here between human and mice cells, and I won't give that away. That's something for you to do some research on. Of course, if you were to choose different transgenic lines here, that's actually a really great experiment. So what you could do is you could choose excitatory and inhibitory lines, for example. Same with spiny and aspiny. So if you want to look at the comparison between spiny and aspiny, excitatory, inhibitory, you'll find a lot of really interesting differences here in the data. Finally, you might want to plot this as a box plot. So here's another way to plot this too. So you could go down here and this will just replot it. Instead of a histogram, it'll plot it as a box plot, which of course gives us a little bit more of a sort of summary of the data, which can nicely be like a final summary plot for students. Okay, so that's all I wanted to show you today. This notebook, as well as the other notebooks, are all on the web resource. You can actually use the same link that we shared with you today to have your students go through this notebook as well. And again, there's a coding light version of the notebook, which has fewer coding tasks. So that might be easier for your students, especially if it's their very, very first introduction to coding. But if you're willing to spend the time to go through this with them, I would definitely recommend that. And it opens up a lot of possibilities for them actually doing their own experiments, analyzing the data in a similar way to the way that the Allen Institute is even analyzing their data, like that they talked about earlier for their publications. So yeah, so I'll leave it there. Thanks so much. And I'm happy to answer any questions.
All right. So if you have questions, please go ahead and keep putting those into the chat here in Zoom or on YouTube. So the first question is actually a great follow-on from the end of the notebook for Ashley, from a user who's new to Google Colab. If students leave this page, does it clear their work?
Yeah, that's a good question. So I believe if you go to File, you can save a version of this in your drive. So if students are logged into their drive, they can actually save a version of the notebook and then reopen it later. So you can give that a shot. Yeah, I think that should work for students.
Great. The next question is for Tom. You had mentioned the patch-seq data. How many neuronal subtypes do you need to pull from to yield high quality reads? I think they are referring to like getting a good distinction between types.
Yes, this is a complicated question, which is why I didn't try to type an answer. I'm not quite sure exactly what is meant here. I mean, so the considerations in being able to distinguish subtypes are, I guess it's really like both how many subtypes we're expecting and how many cells we need to sample. So if we want to reach a finer level of resolution, like more subtypes, we'd need to sample more cells. I think maybe that's what was intended to be asked about. And there I'd say like roughly like 1,000 cells per subtype, or at least like 500 to 1,000 would be like a guideline at the level that we're trying to like pin these down, where we have a lot of controls from like bootstrapping these clustering assignments and making sure we have confidence. So somewhere there. But if that's not exactly the question, feel free to clarify in the chat.
So Tom, you typed an answer to this one, but I think it might be good to elaborate on that. Are all recordings done in current clamp, or do you have additional data? So
yeah, I also want to highlight that one.
Elaborate on that answer.
Yeah, we have another totally different part of the cell types program, not the in vitro single cell characterization, but the synaptic physiology project, which does paired recordings from multiple cells in the same slice. And so they're interested in synaptic response, and so they're doing a lot of both voltage clamp and current clamp recordings of synaptic currents. And those cells have a smaller set of intrinsic protocols as well, so their intrinsic properties can be assessed. And there's some nice Python tools available for looking at that data and processing it. Not quite as easily user-friendly as the stuff we went through today, but it should be pretty good, and we're always improving it and releasing more data there. So really exciting stuff. There's a link in the response to that question. Check out.
And we've just got a clarification on that on the patch-seq question. That they meant patch-seq sample size per cell type to yield sufficient sequencing read, not to distinguish between cell types. I'm not sure about that.
Let's see. Well, so if you're talking about patch-seq, I mean the sequencing is all it's just at the single cell level. So either a cell has enough reads, like the high quality enough extracted nucleus that it works, or it doesn't. And we want it at single cell resolution, so we can't enhance our ability to get those reads by adding multiple cells. But we don't always get enough resolution there. Usually, I'd say it's probably like 20% failure rate, if that gives you some idea. Yeah.
I also see a question that has come in the chat. I think we're going to need a clarification on this one. Is it possible to use the data of single cells and compare it to nerve recordings to estimate what cells are present too? If you can clarify that question, unless Tom, you have you think you know what we're getting at here?
I'm not sure exactly. I mean, I guess they're potentially getting at like electrical properties of neurons outside the brain, in the spinal cord, for instance, or other nerves. I mean, that's something that the Institute hasn't branched out to yet, but if there's other data sources available, I'm sure you could do that sort of thing.
Yeah. So we had a question.
Just a brief response to it. I had a few detailed questions that I answered, like about the electrical protocol. And I did just want to mention there that there is a recent, for people who are experimentalists and more interested in those aspects, well, both to look at our community forums, but also if you're really interested in patch-seq, we just released a preprint of a detailed protocol on the patch-seq process that I'll drop into the chat too.
Great. While you pull that up, we had a question that came up earlier that somebody commented about Coursera offering nice coding resources as well as computational neuroscience classes for students. So Ashley, do you have recommendations of coding resources for students who, it's great to learn to code when you have something to analyze. It really helps to know what exactly the applications of your work is going to be. But what are some good resources for students who need a reference or a starting point?
Yeah, definitely. There's so many free online resources for learning introductory coding. The ones that I like to point my students towards are DataQuest. So that's dataquest.io. And that one has free lessons for most of the basic stuff like assigning variables, even up through NumPy. So those are really great. Also, Code Academy is great as well. And so typically in my class, I have students do 1 or 2 of those introductory lessons before we do it in class, especially for students that feel a little bit nervous about coding. It gives them a chance to try it out on their own, take their time with it, and complete those lessons as well. For more advanced coding, there's also Neuromatch Academy, which is a online summer course that happened this past summer, is really tremendous and was about 3 weeks long. All of their materials are online. You can basically get a certificate in computational neuroscience by working through those lessons.
Great. We have another question for Ashley. I've been meaning to organize my own EFIS data in a database. Actually, this would be for either of you. Do you have any recommendations for building a database from scratch or using available management resources?
Yeah, I think it's a great question. I mean, I think for me, what I would do is I would, maybe start, like, if you have all your data in sort of like an Excel spreadsheet or something like that, that's a great place to start. Those are really easy to import into Pandas and Python for analysis. So that's a great place to start. Tom?
Yeah, I mean, in terms of actual database storage, as opposed to like, yeah, you can get a nice sort of database-like format from Pandas loading CSVs. But like, if you're trying to create like a SQL or other database that could be persistent storage and queried, I'm not sure I would have any clear recommendations. I mean, there's some nice packages that make that easier, like Data Joint, which is particularly neuroscience oriented and integrated with processing of that data, or just at the broader like Python database interface, SQL Alchemy as a way of designing and structuring databases would be worth checking out. But our systems internally are pretty integrated with how we also store and experimental data, and are probably much more complex than like a first step. So I'm not sure I have any super clear recommendations there.
Yeah, one thing I just thought of too is the Neuro Data Without Borders format, which is built for physiology data and is an open access data format and has a lot of really great documentation. So if you have physiology data, so time series data of some sort, the NWB Neuro Data Without Borders format is really, really great for that. And that's actually the format that the Allen Institute data is in as well. And as an aside, they actually have small seed grants. If you're someone who does research and wants to put your data in that format, but may not have the resources, they actually have small seed grants to support researchers to put their data in that format so it can be easily shared with other researchers.
Yeah, you can find that information on the Neurodata Without Borders website, which I believe is neurodatawithoutborders.org. Mm-hmm. So this is our last question that we'll answer live, but as a reminder, we are going to create a post on our community forum, which is community.brain-map.org, where we'll post all of the answers that we've covered during the webinar today. And if you have follow-up questions, you can go ahead and post them there. Or if you're watching the recording of this webinar, not joining us live, and you have questions, we will be keeping an eye on that. So this is not the end of getting questions answered, but this will be our last question for the moment. And this question is for Ashley. Can you do the same types of analysis of spike shapes for different systems like cardiac cells or heart ECGs? And are there studies that you know about this? So a spike is a spike to some degree.
Yeah, I think you could pull out some of the same metrics. I'm definitely not a cardio specialist, so I'm not 100% sure, but probably things like spike width, amplitude, that's still relevant for these heart signals as well. I don't personally know of any open access data for that particularly, but I am often scouring the internet for open access data. And on the website that was linked earlier with the other lesson plans, there's also a big Google spreadsheet of open access data. So if you're looking for other types of data, whether it's like heart data or from EEG or ECOG, that kind of thing, there's a big sort of spreadsheet there with a lot of other data resources because there's a lot of data online.
Well, we are out of time for today. Thank you everyone for joining us. I hope that you will use this lesson plan, explore our open data with your students, and good luck with your teaching both online and in person. Please let us know if you have any questions, and we'll see you at future events. Thank you.
Thank you all.